Very much. Sorry for the delay. So I'm Matthew Scotch from Arizona State University, and I did this work in collaboration with the Mayo Clinic, mostly in Rochester, and then one person was in Florida, and one person was from the Minnesota Department of Health. Let's see. Oops. Did I just click on? Oh, perfect. So far. The work focused on genomic epidemiology, which for those that are familiar, utilizes sequence data to look at the evolution of spread of mostly RNA viruses. If you don't know, there's been a huge growth of sequencing done during the pandemic, with GISAID representing the largest repository of right now about 15.5 million sequences. NCBI is about half that, at about 7. And there's been tons of studies focused on international spread and spread between states and provinces, but much less so on in-state transmission and introductions into the state and how the counties or local regions supported those types of analysis, and then what local health departments could do to combat that. So that was the motivation for our study. So the focus of this was to study the spread within the state of Minnesota, including the introductions from international and domestic sources, then to also look at variants of concern and how they impacted transmission, both pre-VOCs as well as Alpha, Delta, and all the way to Omicron, and then look at statewide decisions in terms of the timing and whether they altered the trends in transmission within the state. So what we did was collected, was a large team effort, collected samples and analyzed them in the Mayo Clinic labs over 2 years, March to March 20 to 22. We only accepted clinical swabs that had CT values of less than 28 or 28 or lower. We did paired and Illumina sequencing and then used Dragon pipeline and excluded those that were of poor quality or duplicates. We analyzed over 22,000 patients. We did a pretty good, for a convenient sample, is pretty close split between males and females. Most of them were between 45 and 64. And then, 18 to 45, with some older, above 45 and 65, and a few adolescents. And then since the Mayo Clinic's located in Minnesota, most of the patients were from that state, a few from the different bordering states there. So because our analysis, which is Bayesian inferencing, relies on a smaller data set than traditional maximum likelihood models, we had to downsample. And with doing so, trying to do a representative proportional by the number of cases. So we selected the top 20 cases, top 20 counties in the state at the time of the analysis, and we tried to proportionally sample at about 5 sequences per 1,000 cases. We used one of the filter method as part of the Augur pipeline to distribute over the 2-year time period as close as possible. And then we created initial phylogenies and looked at potential misassignments in terms of their clades based on the next strain's global phylogenetic tree at the time. So we eliminated those and then redid the analysis. In addition to the number of sequences, we had to discretize the discrete states. So we couldn't have 20 counties in our analysis just because of the Bayesian inferencing, the computational burdens there. So we basically discretized some into northern Minnesota. Whoop, I don't know if I touched it. Northern Minnesota, and then southern Minnesota in the purple. Green is central Minnesota. And then for the 5 largest counties in the state, so Hennepin, which is the orange or red, which is where Minneapolis is, we kept that as Hennepin County, and then Ramsey, which is right next to it, and then Anoka and Dakota. So we kept some, but then discretized the rest. So then here's a look at our distribution. So we downsampled to 6,000, a little over 6,000 sequences. We did a pretty good job over the course of the pandemic in terms of our 2-year timeline with having a consistent number. There's a little dip in the middle there due to just the convenience sample that we had. And towards the end, you can see that most of the samples are international because we stopped sampling at that time, collecting from our patients. So once we had the 6,000, we used MAP for sequence alignment. We trimmed columns that had large number of gaps in them. We used Augur tree to create initial maximum likelihood trees with IQ tree that was rooted based on Wuhan 1. We used Tempest to look at the signal of our heterochronous sequences based on divergence from the root. So that'll look at whether it should be a relaxed or strict molecular clock. And then we used the refined method in Augur to create our final maximum likelihood tree. Because of the Bayesian inferencing that we were using, so this is utilizing a new approach called the thorny tree likelihood, which allows you to do larger data sets in using BEAST, which is the software we use, but it requires a starting tree that's rooted and has polytomies in it. So we wanted to check for that and use BeastGen to create our XML template. So here's a look at our little pipeline flow here. So we're starting with the rooted non-bifurcating tree. We're running two BEAST analysis, two separate BEAST analysis that both implement the thorny likelihood model. We specified a non-parametric population growth through SkyGrid coalescence, and then combined our two Markov chain Monte Carlo runs in long combiner and created a sample of 1,000 posterior trees. We used those 1,000 into a second BEAST XML run that only did ancestral state reconstruction of the migration process along those 10 discrete locations that I just showed you on the map. We also simultaneously counted the number of Markov jumps, which is the number of transitions from one location to another. And then that created our cool little colorful posterior trees here, which then led to our single representative maximum clade credibility tree. So we used both the posterior distribution of trees, and the single MCC tree in our analysis. So we use the MCC tree, we use Baltic, which is a Python library, to extract the introductions from an origin to the destination. And by limiting it only to those with locations of a 0.9 or better posterior probability. And then used two new packages from BEAST that allow you to analyze Markov jump history through the history analyzer, which extracts the timing and location of each Markov jump. And then state time summarizer, which allows you to look at the continuous partitions for each of the 10 discrete locations. And for that, you can do things like looking at the diversity of the virus for each location. So looking at our results, the top is the maximum clade credibility tree. We annotate by color shown in the legend here and on B, they're both the same. And then I show the different variants of concern. So Delta, Omicron, and then Alpha, and then the pre-VOCs. It's hard to see, I know. The red represents Hennepin County. So you can see the dominance there, that it really dominated the spread. And then this is shown through the chord diagram, where the red, the bottom is Hennepin. So it has the Markov jumps going out to it. The white border between that and the location are introductions into each location. So you can see most of them are accepting Hennepin County. There's a little bit of counter from central Minnesota, this blue here, but most of it, as you can see, is just quite massive, the domination of Hennepin, even after proportionally downsampling. Here you can look at the ratio of introductions to total viral flow for each of the different areas of Minnesota. So a 0 means that the location was a source. meaning that it's just basically sending out virus to other locations in the state. One is a sink, meaning it's exclusively accepting virus. So you can see that Hennepin is really the only location here that is anything but a source. As you can see, it's below kind of 0.25 for most of it, a little bit close to 0.5 in the middle there, versus the other locations where it's almost exclusively sinks, except for maybe central Minnesota, which has some source-like characteristics at times. Yep. And then looking at the points of introductions for each location, the colors represent where the source introduction was from. The first introduction into the state was from an international source into Hennepin County in early 2020. And you can see just the dominance of Hennepin with introductions into the other areas. And really, Hennepin was seeded by mostly international locations, and then that just led to the dispersal out to the other counties. And then finally, looking at the Shannon diversity, which is a 0 means you have exclusive spatial structure, meaning the outbreak is from a single location, versus 1 where you have just complete mixture of the different counties. And you can see, despite Hennepin's dominance, most have mild to moderate diversity in terms of where the virus originated from. And then we note with the 2 lines here that this is a policy here in early May 2020, the end of the lockdown in Minnesota, and then in early 2021, the end of all restrictions. And our assumption a priori was that the end of lockdown would lead to a growth in diversity. And we saw a little bit of an increase, but it kind of just leveled off and was what it was at that point. So that was somewhat surprising, and it was fairly consistent, versus the end of all restrictions didn't seem to do much either, potentially maybe as the people were getting the vaccine. And then finally, this is it. Just discussion, we analyzed over 22,000 genomes or sequenced over 22,000 genomes, and then looked at the history and spread within the state, both pre-VOC and then all the way up to early Omicron, identifying Hennepin as the dominant source, but finding limited spatial diversity, and the impact was limited in terms of at least what we saw anecdotally for policy. Limitations, we only capped it at the top 20 counties. So we didn't look at how the smaller populations impacted the spread. We had different versions of Dragon, and then our domestic and international sequences were a convenient sample. So depending on how they were selected, that could change the timing and location of introductions. So just thank you for funding. And then there's our preprint, and happy to take questions. And yeah. Sorry for the delay. Yeah.
I know that you alluded to potential introduction of the vaccine at some point. So first, did you look at any specific regions that had a higher uptake of vaccine rates? Or, I mean, did you look at the county voting records and use that as a potential?
Yeah, I kind of stayed away from that. Yeah. Oh, it was a question about vaccination. Did I look at that as a factor, and or potentially associated factors like voting records? I didn't look at that. I started to explore those types of options. For one, we were going to look at a generalized linear model to help explain predictors, but I was kind of inundated with all the data, so I shied away from that at this point. But it certainly warrants, further explanation. At this point, it's kind of harder to check because, some people have had boosters, some haven't, and even with cases, a lot of people are doing at-home testing now. So some of that stuff was, better to track it early on. But yeah, we didn't do it. That's a good question.